Okay, so we are going to finish chapter 7 today. We will be done with cellular respiration. I know it's been a very long haul in going over, glycolysis, the Krebs cycle, and the electron transport chain. But we are at the end, and all we got to go over today is the anaerobic process of cellular respiration. So in other words, when oxygen is not present, what happens? Okay, so what we're going to do today, guys, is talk about fermentation. And fermentation is the process when oxygen is absent. Okay, so without oxygen, we're still going to have glycolysis. So glycolysis will always occur no matter what. Okay, but without oxygen, we are not going to proceed onto the Krebs cycle and the electron transport chain. So without oxygen, we stop at the end of glycolysis. Okay. So this type of respiration is called anaerobic respiration. In prokaryotes, they still do have an electron transport chain. It's just a little bit different. Theirs occurs in the cell membrane. It's a little bit different than ours. But for anaerobic respiration, we're going to use a different electron acceptor rather than oxygen, maybe like a polyatomic, kind of like sulfate or something like that, okay? But for these types of organisms, guys, it occurs in the plasma membrane instead of the mitochondria because prokaryotic cells, they don't have a mitochondria, right? Okay, two types of fermentation we're going to go over. We're going to go over alcoholic and lactic acid fermentation. I'm not going to go over the whole mechanism. I just want you guys to know the overall goal of fermentation and why it's needed. So we're going to start off with alcoholic fermentation. What we're going to do is we are going to convert our pyruvate into ethanol, which, it's alcohol. We're going to release carbon dioxide, okay, from pyruvate, just like we did in pyruvate oxidation. So we kind of still go through there. The only difference is instead of it continuing on to the Krebs cycle and combining with oxaloacetate, what we're going to do is we're going to take that and we're going to reduce it into ethanol. Okay, this is used by yeast. We make bread with it, and it's involved with a lot of alcohol. Okay, so for this, guys, here's our goal. All right, our goal of fermentation is to allow glycolysis to happen again. Okay, because glycolysis is the only step that these organisms are going to go through. So that's their only means of generating ATP. That's their only means of refueling their energy. So in order for this to happen, this step down here, let me get a different color here. This right here, that is fermentation. Okay? That is fermentation. All that fermentation is in glycolysis, we make NADH, okay? And in making that NADH, we get ATP for energy, okay? We want to continue this process so that we can go through glycolysis again to make more ATP. Sorry, my picture got really messy there. But we want to just keep going through glycolysis. In order to keep going through glycolysis, we need to regenerate our electron acceptor. We need to regenerate our NAD+, okay? So that it can receive the electron once again during glycolysis and form NADH. So all of our goal is for fermentation, is to take our NADH and transform it into NAD+. In other words, lose that electron. Okay, so remember from the first day of this chapter, lose electron is oxidized. All we're trying to do is oxidize NADH to form NAD+, so that way our NAD+ can go back into glycolysis and once again be reduced to NADH. Okay, and here's our two mechanisms of fermentation, guys. Only difference here, I mean, if you look, it's the same exact process. Only difference, Alcoholic fermentation releases carbon dioxide. Lactic acid fermentation, no carbon dioxide is released. All right. And the obvious difference would be in the end of lactic acid fermentation, we get lactate as a byproduct. In alcoholic fermentation, we get ethanol as a byproduct, okay? This is why with alcoholic beverages, whether it's beer or wine or whatever, they are carbonated because CO2 is released, all right? If we were able to make alcoholic beverages with lactic acid, they would not be carbonated. They would not have CO2. Same stuff that's in like soda pop. Okay, so there's the bigger pictures there. We already talked about this with our lactic acid fermentation. What happens? We don't release CO2, okay? For us, whenever we don't have a lot of oxygen available to make our ATP, we will go through lactic acid fermentation. There is a big thing with lactic acid and lactate causing us to get very sore. They're finding out now that it's actually like a evolutionary mechanism where, hey, you're going to get sore, so your body produces this chemical to make you sore, so you stop working out or doing whatever you're doing. Okay, so two contrasting things there. All right, so this is just a comparison between everything, guys. With fermentation, every single time we go through it, we are only going to get 2 ATPs out of it. That's it, because we're only going through glycolysis. With aerobic respiration, meaning we are going through all of cellular respiration, we are going to get roughly, your book says 32 ATPs. It's like 32 to 38, somewhere around there. Now, with anaerobic organisms, we're going to have two different ones. We're going to have obligate ones and facultative ones. And my lovely lights went out, and there we go. Okay. So the first one, they are obligated to go through fermentation no matter what. They're obligate. So they are definitely going to go through fermentation. Their whole lifespan is only glycolysis and fermentation, and that is it. Our second one, yeast and bacteria go through them. This means that if oxygen is available, yeah, sure, they can go through cellular respiration. They can go through Krebs cycle. They can go through the electron transport chain if oxygen is available. If oxygen is not available, no big deal. They'll just go through glycolysis and fermentation and keep repeating that until oxygen becomes available. So they have two different pathways that they can go through, okay? So again, here's the facultative ones, okay? These are not the obligate ones. The obligate ones have to go through the left side here, going through fermentation. Okay, they found that glycolysis is in all three domains. It's in bacteria, it's in archaea, it is in eukarya. All right, this once again points toward us having a common ancestor, since the process is the same in each one of the domains. Okay. They also found that 2.7 billion years ago, guys, there was not a lot of photosynthetic organisms, so we didn't have a readily available oxygen in the biosphere. So given that, these organisms had to find a way to make ATP without using oxygen, and glycolysis was it. All right. Along with this, guys, now we went through the carbohydrate part, that we start off with glucose, and then we are going to take it down into pyruvate for our glycolysis mechanism, okay? We didn't go through all the intermediates because we didn't really need to know them, but I want you guys to know that along with using our carbohydrates, along with using glucose, we are also going to use proteins and also lipids, fats, in order to break down and change our glucose into all the different intermediates it's going through. Okay, so here's just a diagram of everything that happens with our proteins and our fats being used. And this is pretty much all in glycolysis, a little bit in the citric acid cycle, but you can see in glycolysis, we're using the fats to get the 3-glyceraldehyde there, and then the proteins to to get the pyruvate. Okay, so the only one that's really not used here is nucleic acids for cellular respiration, but all the other macromolecules are used. Okay, guys, that is the end of the chapter. We will stop there. There's one more thing there. Sorry about that. Have a good rest of your day. Start studying for your Chapter 7 test on cellular respiration. I'll see you guys later.